Today we finished with ADA deficiency or gene therapy. Gene therapy basically, this is the faulty genes, any faulty or mutated gene is replaced by a normal gene. Basically, this is gene therapy. When the genes they get mutated, what happened that they cannot function properly, and in that case, due to the absence of normal functioning of the gene, whatever protein or whatever enzyme is produced, its production is not there, it's hampered. So in that case, gene therapy is done where that faulty gene is replaced by a normal gene. The example which is given in NCERT book, that is about ADA deficiency. So ADA deficiency causes a disease which is known as SCID. Yesterday we left here severe combined immunodeficiency. The disease's name is severe combined immunodeficiency, and it is due to the lack of enzyme adenosine deaminase. As I told you, ACE is always for an enzyme. So the name of the enzyme is ADA or adenosine deaminase, and the deficiency of adenosine deaminase leads to a disease which is known as SCID. SCID is severe combined immunodeficiency. This is the disease caused by the lack of ADA in the system. Now, what is this? There is a normal gene in the body, and this normal gene produces ADA, this enzyme. If this produces ADA, if this normal gene produces ADA, ADA is produced. This ADA helps in normal functioning of T and B lymphocytes. So when ADA is there, which is released by the normal gene, the T and B lymphocytes, they function properly. And because of the normal functioning of T and B lymphocytes, what happens? The body's immune system functions normally. So body's immune system is strong. The body's immune system is strong, and you know, if the body's immune system is strong, it protects the body from various diseases. So the body's immune system is strong. But in case if this normal gene, if it gets mutated, then what happens? Or if it gets faulty, then what happens? So in the case of disease, SCID, this normal ADA forming gene, this normal gene, it gets defected. So mutated gene. Now we do not have a normal gene, we have a mutated gene. So in the presence of this mutated gene, no ADA is secreted, no ADA is there, no ADA is synthesized. When ADA is not synthesized, there is, you know, the T and B lymphocytes do not function normally. So what happens when they do not function normally? Weak immune system, and the body is susceptible to many diseases. weak immune system, and the body is susceptible to many diseases. And this leads to the disease SCID, severe combined immunodeficiency. So this leads to the disease SCID. Now, the treatment is of course gene therapy. The treatment is of course gene therapy. How this gene therapy is done and where was it done? At first, the gene therapy was done on a girl, 4-year-old girl in the year 1990. In the year 1990, this gene therapy was done on a girl who was at that time 4 years old. So she was suffering from this disease, ADA deficiency disease kid. So she was in 1990, she was 4 years old and she was suffering from SCID, ADA deficiency. And she was given treatment. The first treatment which can be given is enzyme replacement therapy. Enzyme replacement therapy. What is this enzyme replacement therapy? You know, ADA is an enzyme. This ADA is an enzyme, and the name is adenosine deaminase. So this enzyme, ADA, if the person is suffering from SCID or ADA deficiency, that means the person is not having ADA enzyme. So when ADA enzyme is absent, this ADA enzyme can be injected into the body, injected into the body of the person, into the body of the patient. So this ADA enzyme is injected into the body of the patient, and this is the enzyme replacement therapy which is given. The second way, which is gene therapy and which is a biotechnological therapy, how that is done. So in case of gene therapy, which is the second type, gene therapy, what is done? First of all, you know, the lymphocytes from the person as from the patient are taken and cultured. Lymphocytes from patient are taken and cultured. So these lymphocytes are taken and cultured in the lab, okay, amplified in number. So these are taken out and cultured. Then what happens? Then here in this biotechnological method, the use of a retrovirus is there. A retrovirus is used to perform this function of gene therapy. Now what is done? Again in the lab, a normal ADA producing gene is taken. Normal ADA producing gene. This is also taken, and this is introduced in or introduced in retrovirus. Retrovirus is the virus in which enzyme reverse transcriptase is present. That is why we call it retrovirus, because these viruses, they have RNA as genetic material. That is why we call them retrovirus. In retrovirus, what do we have? RNA as genetic material. But you know, when the protein synthesis take place, in that case we have this DNA which forms RNA and then which forms protein. This is a normal central dogma. But in these retrovirus, this RNA is present. So we are at this stage. In the retrovirus are this stage. So from RNA directly protein, no, because this is not messenger RNA. So what is done? A reverse transcription is done. From RNA, first DNA is made and then the complete process takes place. And for this reverse transcription, there is an enzyme which is known as reverse transcriptase that is present. And these retrovirus, they have this enzyme. Now, these retrovirus, which have RNA as their genetic material, these are taken and in them the normal gene is introduced. What do we get? We get this, the normal gene is introduced in the retrovirus. Now this retrovirus, this is also amplified, increased in number, And this retrovirus with the normal gene is allowed to infect. This is allowed to infect the lymphocytes, which are already grown in the culture medium. So these lymphocytes are allowed to infect. Now, what will happen? This is, AIDS virus is also a retrovirus. Now, these retrovirus with the normal gene, this is their nature to infect the lymphocyte. What happens in AIDS? In AIDS, of course, the lymphocytes are only attacked. So when the lymphocytes are attacked, what happens? The immune system becomes weak, and we say that it is AIDS. AIDS has happened to that particular person. Here also, this retrovirus, they are asked to go by their normal nature. So what is their normal nature? To infect lymphocytes. So these retrovirus, which now have the normal ADA producing gene, they infect the lymphocytes. When they infect the lymphocyte, what is introduced in this lymphocyte? Normal ADA producing gene is introduced. Then these lymphocytes with the normal gene, now the retrovirus cannot infect, cannot do cannot produce a disease. Rather, it will do a good job. That is, it will provide a normal ADA producing gene to the lymphocyte. Now this lymphocyte, this way, which have the normal gene, this is increased in number, amplified, and then injected into the patient. This is injected into the patient. Now, this patient which has these lymphocytes have normal gene. That means this is no more patient. This is a normal man. This is a normal man. Because why this is a normal person? This is a normal person because now the normal gene has been injected into this patient, and because of which it has started producing ADA. So this one is a normal patient, normal person now, no more a patient. So this person can now produce ADA, and its immune system also becomes strong. But this is not forever, because the lymphocytes, they have a lifespan. This is not forever. See, this lymphocytes with normal gene has been injected into the person. The person is no more a patient, becomes normal person for a while till the lymphocytes are there. And in this person now ADA is being produced, and the immune system becomes weak. But once the lymphocytes die, lymphocytes, they have a lifespan. After completing their lifespan, they die. So when these lymphocytes, they die, again there is no ADA, and the normal person becomes patient again. There is no DA, no ADA. That means again the disease kid. So time to time the person has to have this injection of lymphocyte with normal gene. If this is not given, again the person comes to the that start suffering from the disease kid. Now, researchers thought that this is not a permanent cure to this disease. There should be a permanent cure. So what they did to find out that permanent cure, they found out that if this particular gene, the lymphocytes with normal gene, if these are injected at embryonic stage. Embryonic stage means blastula, gastrula. Okay, if these are injected at normal stage, at embryonic stage, then it becomes a permanent cure. Okay, then it's not that the person will not have to be given, the periodic injections of lymphocyte. It becomes a permanent cure. So this question I gave you in the test also, I think you all were able to do it. So children, here we finished with the biotechnology in the field of medicine, in which we did insulin, in which we did monoclonal antibodies, and this ADA. So now I switch over to the chapter of ecology, which is left in our syllabus, and that is biodiversity chapter, biodiversity and conservation. I switch over to another chapter, which is biodiversity and conservation. So this is the last chapter of our syllabus. Then we will start revising the syllabus. So the name of the chapter is biodiversity and conservation. What is biodiversity? Biodiversity. What is biodiversity? Biodiversity is the diversity of the life forms found across the earth as a whole or in different areas specifically. Now, this term biodiversity, it was given by Rosen. The term biodiversity was given by Rosen, Rosen. Rosen gave the term biodiversity, and this term was explained by another researcher. The name of the researcher is Edward Wilson. Edward Wilson explained the term, and the term was given by Rosen. Now, as I told you, what is the biodiversity? Bio means life. If we split the word and we understand the meaning, biodiversity. Bio means life forms or life. Diversity is variation. So variation in the life forms or diversity of life forms or variation of life forms. This can be across the earth, found across the earth, as a whole, or in different areas specifically. This is the biodiversity, and conservation, it's protection. Okay. So this is variation of life forms found across the earth as a whole or in different areas specifically. Now, the diversity can be at any level. Diversity can be at the species level, at the tissue level, at the organism level, at the higher levels, or at the lower levels. Now, what are these higher levels or lower levels? Diversity can be at any level of organization. Now, what is this organization? Lower level and higher level of organization. The organization starts from the lowest level, which is, okay, here it is visible. So it starts from macromolecules. Then we have cells. From the cells comes up tissues. From the tissues, organs. From the organs, organ system. From the organ system, organisms. From the organisms, population. Then comes community, etc. I'll write it till here only. I think this is visible in the camera. Macromolecules, cell, tissue, organ system, organism, population, community. Now till the organism, this is lower level of organization. This is lower level of organization. And beyond this, this is higher level of organization. So the variation, the diversity can be at any level. In our book, the diversity which is given here is at three levels: genetic level, species level, and ecological diversity. So it is in the NCERT book, we are given genetic diversity, then we have species diversity, and then we have ecological diversity. How can we differentiate between these three types of diversity? And what are these three types of diversity? Let's understand. Now, we come to the first type of diversity, which is genetic diversity. What is genetic diversity? Here, the example, see, uh, genetic diversity is basically a single species may show a higher level of diversity. Lot of diversity one single species may show. Like if we take example of mango. Mango has more than say about 1,000 strains, various types, various strains, various types. Mango has about 1,000 types. Okay, so this is genetic diversity. It is just one mango and it is present in so many types, about 1,000 types. This is genetic diversity. Another example which is given in NCERT book, this is about the rice. There are about 50,000 genetically different rice varieties. About 50,000 genetically different strains of rice. So see the rice, it is just one crop, which is rice. And see, it is present in a diverse form. And what is that diverse form? About 50,000 strains of rice are present. So rice is showing biodiversity. Rice is showing diversity at genetic level. Another example which is given of genetic diversity is about a plant, Rauwolfia vomitoria. In Himalayas, a plant grows, Rauwolfia vomitoria. Rauwolfia vomitoria. This is a plant which grows in the Himalayan range. Now, this is a medicinal plant and it produces reserpine. It produces a chemical substance, and that chemical substance is known as reserpine or reserpine. So it produces reserpine, and being a medicinal substance, medicinal plant, this reserpine, which is produced by this, is used for various diseases. Now, what is that reserpine? For what disease it is used? This reserpine is used for hypertension and some allergies also. This reserpine is used for hypertension. Basically for hypertension, this is used, reserpine. Now, how does this plant, Rauwolfia vomitoria, shows diversity? It is, you get it along the Himalayan range, but the Rauwolfia vomitoria, which is found at the lower range, and lower range of Himalaya is different from the one which is found at little higher range of Himalaya. And how it is different, how this recipe or how this Rauwolfia vomitoria is different. It is the different in the concentration or potency of reserpine. It may happen that the reserpine which is produced by the plant which grows at the lower range, the concentration is less or the potency is low, whereas the plant Rauwolfia which grows at the higher range of Himalaya, its potency is high, so the concentration is high. It is highly concentrated. You know, you must have seen in hills at Dalhousie also, if you see some plants, they smell a lot, they give a very pungent smell. So that is because, you know, the concentration of the chemical which is present in them is very high. So this is the difference, and you can, this way you can see just one plant, Rauwolfia vomitoria. It can be of various, it can show diversity, and that diversity is in the form of the chemical which is produced by it, and there is difference in its potency as we go from the lower range of Himalaya to the higher range of Himalaya. So this shows genetic variety, genetic variation, genetic diversity in case of this plant, this medicinal plant, which is Rauwolfia vomitoria. We come to the next one, which is species diversity. The next diversity, which is species diversity. So the next species diversity. Now, this is the example of, this is about the greater amphibians. This is about the amphibian diversity, that the variation in the species of amphibians which is found at the Western Ghat and the Eastern Ghat. Now, this example has been given in your NCERT book, and it says that the Western Ghat has a greater amphibian diversity species, greater number of amphibian species. Which Ghats? The Western Ghat. So Western Ghats, they show higher diversity, species diversity of amphibians, and Eastern Ghat, there the species diversity of amphibians is low. So the species diversity, that means you find the number of species at the Eastern Ghat, they are less in number, whereas different species, the diversity of the species which are present at the Western Ghat of amphibians, it is high. So this is the species diversity which is more in the Western Ghat as compared to the Eastern Ghat. Now we come to the next diversity, which is ecological diversity. Ecological diversity. Ecological diversity, that means here we cover a greater area. Now here we take example of two countries, India and Norway. India and Norway. Now, what is the difference between the two? If you see, the species diversity is more in which country, India or Norway? What is your answer? Which country, in which country the biodiversity is more, India or Norway? Yes, yes. Ecosystem diversity. Yes, what do you say? India or Norway? India, yes. Paras Singh says India. That's right. Because in India, what do we get? We get desert ranges, desert ecosystem. Okay, Rajasthan and all. A rainforest ecosystem. What is that? Sundarbans, rainforest ecosystem, desert ecosystem, mangroves also. Mangroves again. This is also Sundarbans. Okay. Besides this, wetlands are also there. Wetlands, Renuka Lake and all that, they come in the wet lake. Then we have estuaries, we have a salt water ecosystem, freshwater ecosystem. We have different type of ecosystem, and the variety, the variation you see in India is more as compared to Norway. So ecological diversity, when we talk about, India shows a higher ecological diversity as compared to Norway because of the presence of different ecosystem. We have deserts, we have rainforest, we have mangroves, wetlands, estuaries, the salty water ecosystem, the freshwater ecosystem. This makes India a ecologically diverse place as compared to Norway. So this is ecological diversity.